Hello, my name is David, and I will be sharing our work titled Alternative Splicing-Based Classification of Heterogeneous Cancers Reveals Novel Disease Subtypes. Large-scale RNA sequencing projects in recent years have become a tremendous resource for modern translational research. An analysis of such data from large patient cohorts has the potential to provide many novel insights into transcriptomic changes that characterize disease subtypes. However, analysis of such data can be challenging due to heterogeneity. Specifically, although all patients in a dataset share a common disease state or phenotype, subsets of patient samples have highly varied clinical outcomes due to differences in underlying genetic abnormalities. Therefore, subtype discovery requires unsupervised stratification of patients into groups characterized by correlated transcriptomic perturbations, which is analogous to finding tile structures in a data matrix. One domain in which this analysis is particularly interesting is subtyping acute myeloid leukemia, or AML, which is an extremely heterogeneous hematopoietic cancer. The traditional paradigm in cancer research has been to analyze genetic mutations as a means to explain disruption of important oncogenic pathways. However, given the low mutation burden in AML, a transcriptomic-centric view, particularly one focused on alternative splicing where exon inclusion or exclusion can significantly perturb protein function, may provide much more valuable insight. To identify splicing-driven AML subtypes, we developed a Bayesian tile finding algorithm called chessboard. Our approach implements a unique probabilistic model tailored for splicing data, which robustly handles missing values and does not require a priori knowledge of the number of tiles. The algorithm takes as input a data matrix in which each row is a splicing event, and the matrix entries X are junction-spanning reads that are mapped to a spliced junction of interest, while entries Y are reads mapped to alternative junctions. This approach enables quantification of splicing as percent splice in, or psi, which is a measure of relative junction usage. The objective of chessboard is to identify a subset of correlated splicing variations which stand out against the background distribution in a subset of patient samples. To achieve this, chessboard pipeline pre-filters the data for splicing variations of interest. Then the Bayesian algorithm is applied to this curated data set and optimized using an MCMC procedure. And finally, the posterior samples are summarized into tile labels for downstream analyses. We first applied chessboard to the beat AML dataset where it recovered a single large tile corresponding to a novel splicing-driven AML subtype. We then applied chessboard to an in-house dataset denoted as PenHTSC to validate our findings and discover the tile is reproducible in this alternate dataset. To further support this reproducibility, we analyzed the correlation of delta psi between the two clusters within both datasets, indicating similar events are differentially spliced. Finally, we trained chessboard on the beat AML dataset and used this model to predict cluster assignments in the PEN dataset, and show that the likelihood of assignment is similar between the PEN and held-out beat AML samples. To explain the mechanism driving this tile, we analyzed whether the tile was enriched for splicing events that are perturbed by misregulated splice factors or RBPs. We assessed whether there was significant overlap between splicing events in the tile and those regulated by each splice factor or RBP, as well as clip binding to local splice sites. Notably, we found that SRSF1 regulates a substantial number of events through direct binding. Next, we recursively clustered the data using only splicing events not included in the tile after each recursive step to discover alternate signals that may correlate with other subtype definitions. In the first step, we discovered a tile containing splicing events which correlated with mutations in FLT3, NPM1, and CEBPA. This is known as the normal karyotype AML subtype. Notably, this clustering is very different from the base case clustering, suggesting that our discovered subtype is splicing driven. To assess the effect of therapeutics on our novel subtype, we reclustered the beat AML data using only splicing events in 70 AML-related genes and discovered similar subtypes. We computed the variance explained by the splicing cluster to various drugs and compared it to the variance explained by mutations. The maximum explanatory power of each mutation was higher than the explanatory power of the splicing clusters. Given that mutations generally predict that drug response better than splicing variations, we sought to answer whether supplementing a mutation-based assay with splicing information could improve predictive power. We found that FLT3 negative patients in the tile respond poorly to sorafenib compared to FLT3 positive patients not belonging to the tile. This new assay, combining mutation and splicing information, increases predictive power for the drug response by twofold. We investigated the functional effect of splicing events in the tile for several AML genes. Notably, for FLT3, patients in the tile tend to produce a non-functional alternative isoform involving exon 4B and exon 17B inclusion, which introduce PTC and frame shifts. For EZH2, we discovered two splicing events which are relatively unchanging in terms of psi between the clusters. However, the tile is enriched for missing values in these events, pointing to the power of chessboard's missing value model. The alternative isoform, including exon 3B and 11, are functionally inactive, suggesting the enriched missingness can supplement the weak psi signal. Finally, we applied chessboard to a joint dataset consisting of beat AML and target pediatric AML samples. The clustering reveals a more complex tile structure. Notably, these tiles contain splicing events in genes which are known to have mutations that are differentially enriched in pediatric and adult AML patient groups. Next, we applied chessboard to the target BAL dataset and recovered 4 tiles. The second tile from the left corresponds to a patient group that is enriched for patients with high relapse rate and lack of RUNX1 ETV6 gene fusion. This gene fusion is generally associated with patients that have positive prognosis. In summary, we discovered a reproducible novel AML subtype that is defined and driven by splicing events. We also showed that combining splicing and mutation information can improve clinical assays for administering therapeutics. In the future, we plan to extend chessboard to include integration of multiomics data, including gene expression and mutations. Currently, chessboard is an open-source Python tool with an accompanying Python package to facilitate data analysis and web-based interactive visualization tool called Gambit. Thank you for listening to this presentation, and I'll be happy to take any questions.